Hello everyone, welcome to week 3 of cell signaling. I'm just having a quick look at some of the culture flasks here, and from one of our groups here there are 12 flasks and we have 2 with contamination. So basically we've got 10 out of 12 that have come through the 2 weeks of the lab without any contamination, so that's actually really good. All right. And for those of you with some contaminated samples, I'm sure we can get some tissue, some cells for you to propagate for next week. Just be kind to those who have cells, and maybe they can just, have a few mils spare. All right. So, firstly, I'll show you what a good one looks like. This is the one that I'm currently working with. I'm pleased to say that there's no contamination. So, if you look here, it's just basically clear solution. All right, so I'll put that one on the microscope. Now, a quick look at the two with contamination. Here's one, and you can see there's a big glob of stuff floating around there. Now, that could be embryo tissue from last week, or it could be contamination. I'm not quite sure. We'll have to talk to the group about that one, but it might not be contamination in that one. And here's one here, and I think this one's, if I can show you, yeah, you can see there's a big mass of stuff there. Now that's clearly not embryo tissue, so that one I think is contaminated. So maybe it's only 1 out of 12 that's contaminated. Alright, so now let's have a quick look at the cells on the microscope. Okay, so we're just having a look at the flask again on the digital microscope. And okay, so we have cells, so I'll just have a look on the other screen. So remember, click here to activate the screen here. Okay, so here are the cells growing on the base of the flask around here. If I move the flask around a little, you can see there's a few things floating around which aren't attached, but most of these cells here, there's a big clump here, are attached and have the right kind of morphology that we expect. So it's a big clump of cells growing here. So pretty much everywhere on the flask we have cells. Okay, so next thing to do is to harvest these cells and do some more culturing of them. So again, record your images of the cells, same as before. Make sure you're in the acquire window, click the acquire button, and then acquire your cells. You need to click this twice before it actually does anything. Okay, so, and here's my image, and somewhere down here it will be listed in the window. So there we go. All right. So take a few snapshots of your cells and record them for your lab book. Okay, here we are back at the sterile cabinet, and as always, clean down your surface before you start to maintain sterility. Now, one thing I'd point out today is to make sure you wear the right glove size. I'm forever sort of looking around at people with the wrong size of glove on, and it does make a difference to how easy it is to manipulate your tubes. Okay, so I've grabbed the medium size gloves here. They've got a size 8, so I'm using mediums. There's a small and there's also a large. Okay. Again, you can touch this region here because when you put the glove on, it folds to be on the inside of the glove, so the outside's still sterile. Okay. Now, there's no loose flaps of latex at the ends of my fingers, and I could go to the small size as well, and they would be equally good. But the large ones are completely wrong because there'd be big flaps of latex flopping around. So here's my cells from last week. We've looked at them under the microscope. This week we'll be subculturing into a 24 well plate. So and you can see here there's 24 small wells. Each one will hold a couple of mils. So we're going to just put, I think it's half a mil of cells into the top half of the plate, and then another group can use the other half of the plate. Alright? And the reason for this is that when the cells are in these small wells, we can treat each cell culture with differently. Okay? Again, we have three medias. We have the trypsin solution to wash the cells off the bottom of the plate. We also have the DMEM for the transferring the cells across, and then we have the growth media so that we'll incubate them in the growth media as we passage the cells for next week. So that's trypsin. So there's the 2 mils of trypsin solution. Again, gentle rinse, and then discard that. And we just leave that for 5 minutes to trypsinize the cells from the bottom of the flask. Okay, so just wait 5 minutes. Alright, so I'm just having another quick look at the cells under the microscope after 5 minutes of trypsin digestion. And you can see the shape of the cells now. They're not stretched out along the base of the plate anymore, but they've lifted and starting to round in up. Okay, so all of these cells that were stretched across the flask are now a much more rounded shape. And right. So let's just have a quick look at some of these. Okay, so there's a couple there still coming off, but most of them have completely rounded. So if we just wash the media over these cells, they should just wash off the base into the next flask. Okay, so you can see they're all kind of rounding up. And you can see here some are floating about a little bit. If I just shake the flask gently, indicating that the trypsin has started to remove them from the base of the flask. All right. So we'll just get a new tube, and just washing a bit of the solution across the bottom here, and then transfer that this is the DMEM. I'm just going to wash 5 mils of this into here. And again, just rinse the bottom of the flask to loosen the cells. Transfer that. I'll keep this and just double check later that I've got all the cells out of it before I throw it away. So we just need to spin these cells down, remove the supernatant, and again subculture them for next week, but this time into the 24 well plate. Okay, so here we are at the centrifuge. Again, I caught some of you last week trying to spin your tubes without using an appropriate balance tube, so make sure you use balance tubes that have the same volume in them and that are diagonally opposite across from the centrifuge. Okay, I'm just having a quick look at my flask on the microscope here, and if you see, if I pan around the bottom of the flask, there's very few cells remaining. Okay, there's just a couple of little spicks and specks here and there, but basically, you know, 99, 95%, whatever, of the cells have been transferred out of this flask into the new, you know, for propagating. So you feel free to just double check that if you're in any doubt whether your trypsinization has worked. All right. Alright, so here are the cells we've just centrifuged, and you can clearly see there's a small pellet at the bottom of the tube there. Hopefully you can see that. So I'll just decant the solution away. It doesn't contain the cells. And there's my cells there at the bottom of the tube. Right. Now, just take a mil of the growth media. And add that to my cells. Just gently resuspend that. It's probably hard for you to see, but I've straight away seen my cells resuspend quickly and easily, unlike last week where they were a bit more compacted. So that's kind of nice. So I'll just take 0.2 of a mil from these cells for counting, and then we will transfer the rest into the 96 volt plate here. Again, it's important to resuspend your cells properly, so just a quick vortex on the vortex mixer should be enough. Alright, so again, just take a small aliquot out for the counting. Again, about just more than 100 microliters, and then keep that for later. And then we need to add a larger volume to our cells now. We need to make our cells up to about 6 mils, because we're going to add half a mil to each of these wells here. So we need about 6 mils of media. So probably do that by eye. You can see we're sitting at about just over a mil, so make that up to about 6 mils. Again, you need to mix that fairly well. Maybe it's best to mix that more vigorously. Try not to wash the solution up near the thread of the tube, because it's the thread of the tube that's being opened and closed, and if you're going to get contamination, it's going to be around the lip of the tube here. Alright, so here's our, oh, good noise, here's our flask, and just 1/2 ml into each. So I assume it's about 3 mils there, so okay, so now unfortunately I've only prepared 10 of my 12. I was a bit quick and rough with the measurements there, but basically we have a series of treatments for next week, and you need at least 8 of these samples, I think, because you've got 4 treatments and you can do them in duplicate. So aim for 12, but get at least you need at least 8. So again, we'll label this and we'll put it in the flask for next week, and then we'll do our treatments on the individual wells. Again, you need to count the cells using the hemocytometer and the microscopes on your benchtop, and then we can hopefully wrap up fairly early this week, because the past 2 weeks have been quite long weeks. So let's try and wrap this one up quickly. Alright, so good luck, and yeah, things are going well, so